start? All right, cool. Should I start? Yeah. All right. Hi, everybody. Thanks for having me here. It's a real honor. My name is Omer Weisbrod. I'm a postdoc with Alkes Price at this Harvard School of Public Health. And today I will be speaking about using epigenetic data for GWAS interpretation. All right, so let's go. So I'm going to start kind of in a light way by first saying a few words about GWAS. So GWAS are genome-wide association studies, and we have lots of genome-wide association studies. So the idea is conceptually simple. We collect a large sample of individuals, and we genotype them, and we measure some phenotype or a bunch of phenotypes, and then we look for associations between these genotypes and the phenotypes. And here you can see the number of GWASs that we have between 2007 and 2017. So you can see that we have lots of GWASs, and the number keeps increasing from year to year. And you should pay special attention to these dark blue bars, which are the huge GWASs with over 100,000 people. So we get more and more of these GWASs year over year. So there are a lot of GWASs, there are lots of genetic data out there. And what do we learn from these GWASs? So one of the main things that we've learned so far is that, sorry, before saying that, I should also mention that not only do we have these GWASs, we also now are in the age of biobanks, and biobanks are these huge samples that are collected by countries or country-like entities. And the largest sample, largest biobank that we have to date is called the UK Biobank, which you may have heard about. So they collected genotyped and phenotyped around 500,000 people from the UK. So this is close to 1% of the UK population. And the data is relatively easy to access to researchers. So this is an amazing resource. So here you can see a map of the UK, and every dot here is the place of birth of one individual in this cohort. So we have lots and lots of genetic data. And so what do we learn from all this data? So one of the main things that we've learned is that most traits are really polygenic, meaning that they are affected by multiple variants, not just one. So when you read in the news that scientists found the, I don't know, the gene for height or for blood pressure or something, it's probably not true, because there is no one gene for anything. Everything is very polygenic. And to illustrate that, I'm going to show some results from a Twitter bot called GWASbot. So GWASbot is a Twitter bot that posts the results of one GWAS per day. You can follow it on Twitter, and just a few examples. So let's look at this example first. So GWASbot posts plots like this. So this is the results for a GWAS for blood pressure medication. And all right, so what can you see here? So on the x-axis, you can see genetic variants in the genome, or what I'm going to call SNPs from now on. So every dot like this is a SNP, and they're arranged according to their physical location on the genome in chromosomes, so chromosome 1, chromosome 2. And on the y-axis, you can see minus log 10 p-value of the statistical tests that ask whether some SNP is statistically associated with the phenotype. And you can see this red dashed line here. So everything above this red dashed line is statistically significant after multiple testing correction. And we impose a pretty stringent multiple testing correction because we test lots of different hypotheses. We assume that we have around 1 million effective SNPs, and so we correct for 1 million different statistical tests. So everything above the red line is statistically significant after multiple testing correction for 1 million hypotheses. And you can see that we have lots and lots and lots of SNPs that are statistically significant even after this very stringent multiple testing correction, right? And so this is not unique. This is what we typically see. So I'm going to show you a few more examples just to get you in the mood. Miserableness. So this is GWAS for the property of being miserable, according to a survey filled by the participants of the UK Biobank. You can see that we have lots of signal everywhere, and everything appears to have some genetic component, polygenic genetic component. So just two more light examples. Hot drink temperature is very, very polygenic. So some people like their temperature, their tea hot, and it's affected by genetics and cereal type. So basically everything. So this is just to get you in the mood. All right, so hopefully I convinced you that lots of stuff is genetic and polygenic. And the question is, what do we do with all these SNPs that we found? Because if we would have found, only one SNP in one gene, then we would simply try to understand what's going on in that gene, and that would be simple. But it's not as simple as that. So it turns out that there is a pretty limited role for protein coding regions in general in GWAS associations. So here are results that were published over 10 years ago already, but the main theme still stands, that protein coding regions play a limited role. So that was in 2009. There is something called the GWAS catalog. Back then it had only 465 GWAS associated SNPs. Now we already have probably tens of thousands. And back then they saw that only 11% of GWAS associated SNPs lie in coding regions. So coding regions account for 1% of the genome. So is that good or bad news? So on the one hand, you might consider it as bad news because it means that 89% of the GWAS associated SNPs are not in genes, are not in coding regions, and that makes interpretation much more difficult. We don't know what exactly they do. They probably regulate some other genes in some way, but it becomes complicated, right? So that's one thing. And the good news is that it's 11%, not 1%. So 1% means that these coding regions are completely irrelevant, right? So it means that the null hypothesis holds that there is no special role for these coding regions. So in fact, we have 11-fold enrichment. And if you dive deeper, they saw that 9% of these 11% were non-synonymous SNPs. So again, that confirms what we know about biology, that non-synonymous SNPs are important and they affect trait biology more than other SNPs. So it's both good and bad because we still have a lot of genetic signal coming from non-coding regions, and we don't really know how to interpret that. And so in order to help us interpret these results, we often use what we call functional annotations. So to understand what this means, here is a figure that I really like from a 2014 paper. And what you can see here is, all right, so you have this green blob here. So these are the set of SNPs that are actually affect some phenotype or the set of phenotypes that we care about. And let me pull the zoom window. All right, because now we can see. All right, cool. So, and we have multiple sources of evidence that could help us understand what these SNPs in the green blob are. So we have the set of protein coding SNPs or genes, so the set of SNPs in coding regions, which I already mentioned. And we have another source of evidence, this red circle, which are SNPs with evolutionary evidence. So for example, SNPs that are conserved in mammals, if they're conserved or if they're in regions that are relatively conserved in mammals, that means that these regions probably play an important biological role, right? And that's why they're conserved, because of natural or of negative selection, right? So that's another source of evidence. And then we have SNPs that have that there is evidence that they participate in biochemical processes in the cell, meaning that they probably regulate some genes or participate in the transcription process somehow. And in this figure, they stratified this set of SNPs into 3 levels of confidence: high, medium, and low. And you can see that the green blob intersects with all of these other annotations. So this gives you kind of an idea of how we can think about these functional annotations. And now I'm going to very briefly go over the types of functional annotations that I'm going to speak about for most of this talk. Not all of them, but very briefly. What are the types of functional annotations that we often think about? So first we have genomic region, whether a SNP is in a coding region or in a promoter or stuff like that. And chromatin state is a very important functional annotation because we know that SNPs in open chromatin regions are more likely to be involved in biological processes. And so we have information coming from DHS, DNA's hypersensitive tie sites, or predictions based on histone marks. And we can also think about histone marks as an annotation in a very explicit way, so stuff like methylation or acetylation. So this is another source of evidence that a SNP plays a role in some biological process. And we have regions that are evolutionary conserved. And so these are probably important SNPs. And enhancers or super enhancers, these clusters of enhancers, which increase the likelihood of a transcription factor binding to a gene. And we have a few others, which I will mention later on, but these are the main ones that I'm going to speak about. And I should mention that this data, a lot of it comes from two very big consortia called ENCODE and Roadmap. So these are amazing, amazing consortia which collected lots of these functional annotations and measured them. And even though the papers are a few years old, most of the data that we use still come from these papers. All right, so these are our functional annotations. And now let's speak about what we can use these functional annotations for. And so I'm going to speak in this talk about 3.5 different tasks that we can do with these functional annotations. So the first is understanding genetic architecture, and I'll explain what this means later on. And a kind of a sub-goal of that is identifying disease-relevant tissues and cell types. So if there is some disease, we want to ask what are the most relevant cell types that drive this disease. So that's one task, or 1.5. The second task is identifying causal variants, which variants are causal for some disease or trait. And the third is genetic risk prediction, so trying to predict phenotype or trying to predict risk of some disease based on genotypes. And I'm going to show you how these functional annotations could help us perform all of these tasks. And before diving into the nitty-gritty details, I just want to mention that everything that we're going to speak about in this lecture is going to be based on polygenic models. So I already tried to convince you that almost all traits were polygenic. That's what you can see here in the bottom right, sorry. In fact, we get more and more evidence that everything is polygenic. So there is a very influential paper, perspective paper, that was published almost 3 years ago, saying that maybe we should not use the term polygenic, but use the term omnigenic. And this paper made a claim that almost every gene probably affects almost every trait. And that's because of gene-gene network. So there's kind of a butterfly effect where if there is a variant in one gene, so because of gene-gene networks, the effect will propagate to other genes and will eventually propagate to all of the directly causal genes, meaning that every gene is at least indirectly causal for each and every trait. And it's a controversial paper, but the bottom line is that we think that most traits are very polygenic. And so in this lecture, instead of looking only at these genome-wide significant SNPs, the SNPs that are above the red line here, for which we typically have at most hundreds of SNPs for a typical trait nowadays, we're going to simply look at all SNPs, millions of SNPs, and not even make the distinction whether we think a SNP is significant or not. And I go into more details later. And I want to mention two very important concepts for the rest of this talk. So the first concept is SNP heritability. SNP heritability is defined as the proportion of trait variants that can be causally explained by all SNPs in the genome. And this is an important quantity. This basically asks how genetic a trait is, right? How much is it influenced by genetics? Because we know that some traits like height are extremely genetic, and other traits like, for example, BMI or cholesterol levels are less genetic. So that's SNP heritability. And the second concept is functional enrichment of an annotation. So it's defined as follows. So it's a ratio. On the numerator, you have the proportion of trait variants that is explained by SNPs in some specific annotation, maybe, for example, by SNPs in coding regions. That's our annotation, maybe. And on the denominator, we ask what is the denominator, what is the proportion of SNPs that are in this annotation. So for example, for coding SNPs, we know that the denominator is 1%. I already told you that. And then the question is, what's the value on the numerator for some trait? If it's large, then that means that SNPs in coding regions are functionally enriched for that specific trait. And this gives us a clue into the underlying genetic architecture. If we can find for some trait what are the most important functional annotations, then we'll know maybe where to look and how to try to understand their underlying biology. And I'm going to show you some specific examples later on. All right, so that's, Yes, sure.
Because I think it's a good time to stop the recording and restart again. It's
Yeah.
a new place. Do people have questions?
Sure.
Okay, I think it's very clear.
I do have a question.
Okay.
Sorry.
Sure.
But can you go back to your last slide? Like,
Yeah.
I'm not sure, like, what do you mean by the denominator, like percent of SNPs that are in the annotation?
Among all SNPs that are in the genome. So if you look at all the SNPs in the genome, how many of these are in the specific annotation that we're looking at right now?
Oh, yeah. Oh, okay.
Yeah, so for coding SNPs, it's approximately 1%.
Okay, great. I'm gonna stop the recording.